Welcome, everyone. We're going to allow for a few minutes so everyone can join. Okay, it looks like everybody's starting to come on into the room, so we're going to go ahead and get started. Welcome, MPN Research Advocates. We are thrilled to have you here for session 8 of the MPN Pathway series. So for any new participants, I want to say how happy we are to have you join the MPN Pathways program. My name is Sarah Douglas, and I'm the Director of Patient Engagement at the MPN Research Foundation. We really want you to all feel comfortable reaching out to us as you're going through this journey with the MPN Pathways. So any questions you have along the way, please, we can be reached at patientengagement@mpnrf.org. So this slide here outlines the remaining schedule for the MPN Pathways Empowered Voices in Research program, designed to help patients and caregivers better engage with scientific research in the MPN space. Today we are presenting session 8, and this is Patients and Caregivers Guides to Scientific Presentations, led by Dr. Tyler Parsons and Dr. Amiel Moreno. So this session will focus on building confidence and understanding and navigating science content, especially helpful for those who are new to research settings. On July 27th, we will move into patient-reported outcomes and quality of life in NPNs, presented by Dr. Ruben Mesa. So this session will highlight the importance of incorporating the patient voice into research with a focus on quality of life and real-world experiences. And then finally in September, we're very excited about this for Blood Cancer Awareness Month. Participants will have the opportunity to complete a certification and interest survey. So we will recognize your participation in helping guide future engagement opportunities. Individuals must complete all 9 evaluations to be certified at the end. So please reach out if you have any questions on maybe which sessions you still need to complete. But you can always find more information and stay connected on our website, and that webpage is listed at the bottom of the screen there. We want to definitely take a moment to recognize and thank our MPN Pathways sponsors and collaborators for their support of this program. For our global participants out there, on this slide, I'm going to walk you through how to turn on Zoom's real-time translation captions. So this feature allows you to view captions in the language of your choice, even if the presenter is speaking in a different language. So first, start by looking at the toolbar at the bottom of your Zoom screen. For step 1, you may either see a More button, that's the icon with the 3 dots, or a Show Captions button. So if you see the More button, click it and select Captions and Translate. If you see Show Captions, click that to turn the captions on. But after that, look for a small settings arrow next to the Caption button where you can access those translation options. For step 2, turn on the translation feature by clicking the toggle button on the right side. When it's activated, the color will change to blue. And then next in step 3, under My Speaking Language, click the arrow and select the speaking language from the list of options there. And step 4, My Caption Language, click the arrow to select your language you want the captions to appear in. And once you've chosen those languages, Zoom automatically will generate and translate the captions for you in real time. Some more housekeeping items. So for tonight's presentation, we encourage you to place questions in the Q&A feature during the presentation. Our team will be answering questions as the speakers present, but please only utilize the chat feature to communicate with one another. We encourage you to introduce yourself and say where you're from, because we think that really builds community. So take a moment to do that as we're starting the webinar. But questions can be missed by our panelists if they're not in the Q&A feature. So please make sure that you're doing any questions regarding the presentation there. If there's time at the end, which I think there will be today, we're going to ask questions of our speakers live, and we'll take advantage of that at the end of the presentation. But any questions that are not addressed will be followed up with an email in a few weeks. Also, as a reminder, we do apologize, but during this session, we are unable to answer really any personal questions related to your direct care or your treatment of your MPN. We encourage all participants to email us, again, at patientengagement@mpnrf.org, so we can help you navigate to the right resources and education. We can help you with questions you can ask your provider or help you find an MPN specialist closest to you. Tonight's presentation is being recorded. That link will be provided to the MPN Pathways group after the webinar in a post-event email. The evaluation of this session will be sent to you in the post-event email as well. Please be sure to complete the evaluation for this session to earn credit towards your certification. And finally, next, I'd like to introduce our speakers who will be guiding you through this program. First, we have Dr. Amiel Moreno, who serves as the Scientific Content Manager at the MPN Research Foundation. Dr. Moreno has a doctorate in neuroscience from Emory University and has dedicated her career to science, communication, journalism, and academic writing and editing. Dr. Moreno's work focuses on translating complex science research into clear, accessible information and making it easier for patients and caregivers to understand and engage with research. And then also, we're very lucky to have Dr. Tyler Parsons join us tonight. Dr. Parsons is an MPN researcher at Washington University School of Medicine in St. Louis, and he is one of our valued members of the MPN Research Foundation Patient Impact Council. His combined experience allows him to bridge the gap between scientific research and real-world patient understanding, helping ensure research is both meaningful and relevant to the patient community. Together, these speakers bring a powerful combination of expertise and lived experience that will support you throughout this program. And with that, I'm going to ask Dr. Moreno to come on camera and take it away. Thank you.
Hello, and welcome to Pathways. My name is Amiel Moreno, and today Dr. Tyler Parsons and I will take you through a guide to scientific presentations, making you a better audience member. Oh, excuse me, I'm learning how to advance the slides. First, let me reassure you that in science, confusion is normal. Science is all about trying to make sense of incomplete puzzles. It's important to remember that feeling overwhelmed is not a personal failure. Today, we're going to teach you what to expect from science presentations. We'll explain where science is presented and about the different kinds of presentations. We'll learn the common structure or patterns to science talks, types of data and studies, and how to interpret findings. All of this will prepare you to identify the key conclusions scientists are trying to communicate. To begin, let's discuss why scientific presentations can be so challenging. Let's talk about knowledge. I want to share something that my first principal investigator taught me, or my first science boss. Let's pretend that this circle is all of the scientific knowledge known to humans. Out of high school, you're lucky if you understand this much of the larger circle. Now, let's say you go to college and you learn more, and maybe you major in a scientific discipline, gaining knowledge in a specific direction. Some people, like Dr. Parsons and I, decided we wanted to know more, so we went for our doctorate. And that knowledge is represented here by the red area. The hope and dream of every doctorate student is to receive your PhD, and they only give you one of those if you press and expand that larger circle of knowledge, poking through to discover something new. All of this to say is that a lot of scientific presentations are explaining that tiny red point of new knowledge and the cutting edge research that could push our understanding further. Researchers presenting at a conference or seminars are usually speaking to people who already share a deep background in the field, people in that red area of the circle. That shared background lets presenters move fast and skip over foundational explanations. Many scientific talks and papers aren't meant to teach a topic from the beginning. They're designed to quickly highlight what's new, a new idea, a new method, groundbreaking results, or conclusions, and it's all for an expert audience. These presentations are optimized for peer evaluation, not first-time learners. So scientists are listening for things like, is the hypothesis reasonable? Are the methods sound? Are the results statistically convincing? And what are the strengths and weaknesses of that experimental design? That's why you often see many figures and very little time spent on context. For patients and caregivers, this can feel overwhelming because presentations, they simply weren't built for you as the primary audience. What specifically makes scientific presentations hard to follow? One major barrier is acronyms and jargon. Researchers often use shortcuts, terms defined once, or sometimes not at all, because they assume everyone in the room already knows them. Another challenge is density. Months or even years of work may be compressed into just a few minutes, with multiple experiments and results shown in a single slide. And as I mentioned before, there's often missing context. Presenters might not spend time explaining why the study was done, what came before it, or how it fits into a larger picture. All of this creates high cognitive load. You're processing unfamiliar terms, there's complex visuals and statistics, and rapid explanations all at the same time. And if you miss an earlier concept, it can be very hard or impossible to catch up. The key takeaway here is this: difficulty following a scientific talk doesn't mean you're failing. It just reflects the nature of many presentations. I want to pause here and say something clearly and explicitly. You're allowed to understand the key conclusions of a scientific presentation without understanding everything in the science presentation. These aren't exams and you're not being tested, even though it feels like you're back in school and you really care, you want to know. But it's completely okay to miss details, unfamiliar terms, or whole sections of the presentation. You can still walk away with the key conclusions. If you understand what the speaker is trying to show and why it matters, you've done a fabulous job as an audience member. That mindset alone can take a lot of pressure off, and that's very important. Let's review where and what type of scientific presentations you may encounter. There are beginner and expert level spaces for scientific discourse. You can identify beginner level spaces because of the comfortable amount of background, which might even feel like review. Clearly defined terms, reminders of why important things matter, fewer figures or terminology, all of which is designed to feel approachable, friendly. And as I mentioned previously, expert level spaces feature presentations designed to share new and novel findings instead of explaining what they think everybody already knows. This means that rationales and methods might be compressed to spend more time on those findings. So here's an important thing. How can you quickly tell the difference? Well, I found that who is presenting is less important than what they're presenting. I don't know what type of talk it's going to be until I read the title of the talk. The tip is the more jargon in the title, the more technical the talk. There's also some other tells. There's the amount of acronyms or how many figures they're presenting and the overall pace of the talk. But guess what? Unfortunately, sometimes you're not going to know that until you're already in the presentation. That's why a good mindset is so important. So this is my most boring slide. You'll find science presented in lots of different ways. You can find it in media, print, or online. For beginners, this looks like health news coverage or popular science magazines. For experts, there are biotech news outlets or pharma companies putting out press releases. The biggest source of scientific truth is scientific journals that put out peer-reviewed articles. And in clinical spaces, there are regulatory meetings and governing bodies that release documents for physicians. Webinars can be live or recorded. They're either for beginner or expert audiences, featuring accessible content on healthy living for patients or how to diagnose and treat patients for clinical audiences. And finally, conferences offer in-person or virtual attendance. You can think of them as adult science fairs hosted by professional organizations. Conferences often feature highly technical presentations that rapidly share new information, followed by intense question-answer sessions. These spaces are perhaps the least beginner-friendly, but we're seeing an increasing amount of beginner and advocacy spaces at conferences and in all of these other formats. Moving on to types of presentations. In order to present at a conference, participants submit a short description of their study to be reviewed by the conference organization. If selected, then the participants present their experimental findings in a poster or oral presentation. Posters are literal paper posters. They're set up in long rows, and one of the authors stands next to their poster, and the attendees get to ask to be taken through a poster talk. A poster has a condensed summary of the experiments or clinical trial, figures, and findings. Sometimes posters are cited by other science content because they did receive some level of review. If the poster submission is impressive enough, the conference might upgrade it to an oral presentation. This is considered prestigious. An author will give their poster talk in a slideshow format to an auditorium audience. When the researcher's findings are mature enough, they'll group them all together in a manuscript for publication in a scientific journal. Science peers will review the experimental design and the findings, suggesting additional experiments. And if everything turns out, they will hopefully greenlight the manuscript to become a scientific article in a peer-reviewed journal. Scientific articles are the most reputable piece of science content. So short of doing all of the experiments themselves, fellow scientists in the field are providing a stamp of approval. Papers are often cited as evidence. All of this science, all of these science presentations share the same building block of science communication called an abstract. The word abstract comes from the Latin word abstractus, which means drawn away. So in art and literature, abstract is an adjective that means drawn away from reality. But in science, abstract is a noun that means a collection of key ideas drawn away from a larger body of work. So abstracts are not abstract, but they are abstracts. They are short summary paragraphs which contain all the information the authors of a poster, oral presentation, or a scientific article feel you need to know about their study and findings. They will always contain the primary statistical results of the experiment. Here's the first page of a peer-reviewed science publication. So as you can see, following the paper's title and the authors, you can find the abstract right before the introduction/background section. And to describe the various sections of a science presentation, I'm going to hand it over to Dr. Tyler Parsons.
Hello, everyone. And again, thank you so much for joining us tonight. I am going to walk through two separate sections that I've been given. I think the first is exactly what was just presented, but in more of a piece by piece of what you might see in a presentation, whether that be an oral presentation or a poster presentation. All of those would consider putting each of these different sections into them. And then the next section is going to be a little more challenging. I am trying to walk through different types of figures that you would likely see in an oral presentation or a poster presentation, specifically at an MPN related presentation, and we'll try and walk through those and The first couple are very common, and then we get into things that you see a little more as we get doing more sequencing with patient samples, and we'll try to walk through, and hopefully you can walk away with a little better understanding of how to analyze those as they're being spoken at a presentation. Let me try. Okay. So the core components of a scientific presentation, this is kind of muddy, kind of gray. I broke it down into six because having three on each of the next two slides was the easiest way to do this. But you could make an argument that there's a couple more sections that should be in every presentation. But of course, you have a title and a background, the hypothesis, which is very important for uh, not only exactly what it is and what you all know it is, but also as a line of kind of demarcation in a presentation where we are moving from things that are known into testing all of the things that that particular study set out to test. And then you have your methods or the way that they did things, the results, and then the discussion slash conclusion. So I have these separated three and three, and the first part is the opening. And of course, this goal is to capture the audience's attention. This is done with the title, the background, and the hypothesis. The title should be short and clear, and this is likely if many of you have attended a conference or watch online, pretty much exactly from the title, you decide where you're going to go during that conference. The titles should be pretty clear as to what the finding was. And I know a lot of patients sometimes ask, how do we know if it is a review article or an original research article? And the way that I can kind of just tell is from the title. If it is kind of funny or it's a pun, probably that title is suggesting that it's a review article, which is just a summation of known work in a nice condensed way to be able to see everything that we know currently about MPN PDX models. That would be a review article. But if the title is more a finding, elevated JAK2 V617F VAF is correlated to higher fibrosis in myelofibrosis patients. That's a title. That's also the main finding of the paper. The next section is the background, and that is why was the study done? And this is where the authors will describe what is known. And as again, you are kind of following along as the presenter is telling the story of their work. It should start kind of big with a funnel and get closer and closer and closer down to the hypothesis. Usually, there's a sentence in there about, therefore, we asked the following. Or in summation, we know this, but there is not a clear understanding of XYZ. And that's usually language suggesting you're about to arrive at the hypothesis, which is what is the study's testable claim? This is a statement. A good hypothesis is never a question ending in a question mark, but it's a statement. So going back to the title example, an example of a hypothesis for that study could be JAK2 V617F, elevated VAFs end in higher fibrosis. That is testable. There is a lot of ways that you can go about trying to answer that question, but that's your hypothesis. It's a testable claim. And I have here in the bottom left of the slide in a 10-minute talk, which Dr. Moreno was talking about oral presentations. Most of those are about 10 minutes, unless you're giving a plenary. And in a 10-minute talk, the first half, the first three sections of the six sessions should be less than 2 minutes long because you really want to save time for the next part, which is where you have 8 minutes to clearly convey the results and the impact of your work to get the audience to buy in to what you are saying for them to walk away maybe with more questions, which is great, or maybe they came with a very specific question and you've answered it. But this is your time can that you can that you can try to answer all that in these last three, what I call the main slides. So there's the methods, the results, and the conclusion. So in the methods, this was how the hypothesis was tested. And that's the description of models. It should be very clear what cell types were used, what patient types were used, what samples were used. It should be very clear how the outcome was measured. When section 4 is muddy or gray, it does begin to pique a little bit of questions with the actual results, the findings, and the conclusion, if it wasn't very easy to follow what they did. The results is what was found. It also gets a little gray and muddy if in the results section you hear the presenter start positing about what it could mean. We have cured something. This has these incredible results, or the first time anyone in the world has shown these things. None of those statements should be in the results. The results is just very black and white. This is what was found. This is the data. And you can have a concise summation of the data patterns. That's fine. That's not really having any kind of discussion injected into the results. But once you get to the conclusion and why do the results matter, now you're getting into why the presenter or the author thinks that those results matter. And this is where sometimes people in the audience might differ in their opinion. The results should be, like I said, black and white. Everyone walks away looking at the results, the same results. The conclusions and the discussion is where things can get maybe a little more gray with, well, maybe it's not as good as what the authors were suggesting. Or actually, I think they're underselling this. That kind of stuff should come in the conclusion and the discussion. I tried to come up with some key ways that data is shown in MPN research and walk you through them to try to demystify them. They get a little more mystifying the further down this list we go, but starting with bar charts and box plots, and I'm going to give examples of each of these on the next couple slides as well, but just quickly what these different visuals do and represent. The box charts and bar charts, they just compare groups side by side, and they should show the range of values. Pretty easy. Everyone has seen these. Scatter plots are always on an X and Y axis, and they show individual patient or sample results. Survival curves, which I know everyone has seen as well, show how outcomes change over time in different cohorts. And then we're getting into a little more unique ways that scientists show data that might not come up in other careers or fields. Volcano plots and heat maps highlight the largest and most significant changes. And again, I'll try to walk through some examples of these so that you understand them better when someone's presenting them, because a lot of times, especially for volcano plots and heat maps, there's kind of an understanding that the audience already knows exactly what they're looking at, and there's not a lot of explanation given. And then for UMaps, these cluster similar data points into a map shape. And as I said, this is getting a little more mystified the further down this list we go. And UMaps definitely are, even to us scientists, probably one of the most mystifying ways to show data. And I'll explain that why in once we get to that slide. So the last thing that I wanted to do before showing examples of those was just quickly walk through all of the things that you're going to see on every kind of plot. And replicates, this is pretty self-explanatory, but I did want to spend a second to define technical and biological replicates, because these are terms that you will hear, and they do have different power. So technical replicates are the same sample repeated. So say I took blood from a patient and I ran that sample from that patient 3 times. I did get 3 different data points, but that was all from the same patient. So that's a technical replicate. A biological replicate is I take blood from 3 different patients and run the same test. I still have 3 data points, but this is a little more powerful because it's from 3 different people. A lot of times, scientists will take technical replicates of each biological replicate. So I could take 3 blood draws from one patient, 3 blood draws from a second patient, 3 blood draws from a third patient. And there's ways that you can normalize that data to, at the end, still show just the 3 biological replicates, but you've gotten rid of maybe some technical error by doing that and giving maybe a more representative figure of the data. This is always represented as n equals a number. So for both of those, n would equal 3. But of course, that 3 is doing a lot more work in the biological replicates than it is the technical replicates. And then moving forward to significance, you'll see asterisks all over data presentations. This is just that The chance that it is purely based on error or chance is greater or less than 95%, and that is usually represented as a p-value of 0.05. Sometimes that 0.05 is not listed. One asterisk is kind of understood to be 0.05. Two asterisks, three asterisks. The more asterisks you have, the more significant it is. There's not really a standard for what all the extra asterisks mean, but for one, it's almost always 0.05. And then lastly is error. And you will see this again in every single way that these are presented, any kind of way that data is presented, especially when there's replicates. And this is those kind of the bars that go on either side of the data. This is just the natural discrepancy in observed values. And it's actually very odd and off-putting if there are no error bars in the data, no matter how bad they might be. If someone is showing just the average of something and there's no error bars, typically that means they're probably trying to hide something or they didn't get good advice for how to make a presentation. So I'm going to walk through just quickly examples of each of those types of data and kind of what you're looking at, so you can quickly be able to pull away what the presenter is saying. Like Dr. Moreno said, a lot of times we talk fast. We're trying to show off all the work that we did in a 10-minute talk, and a lot of times explanations for how to read these things are absolutely not clear. So bar charts and box plots, again, they just compare groups side by side. They clearly show their range of values and the individual data points are preferred. More and more when you publish now, if you submit that first version of a manuscript, they're going to come back and say, hey, in your bar charts, you didn't put all the individual points, please add them. So on that top row there, you can see you have the bars, which of course where the top of the bar is the average or the mean, and then all the individual data points. Box plots on the bottom, pretty similar, but instead of going all the way down to zero, they are kind of more clustered. And that line in the middle can either be the mean or the median. So you can see all the dots that are above the mean, all the dots that are below the median. And then I also have in the bottom right just an example. Sometimes they're called violin plots. It's just a box plot, but it has a little more shape to it. And it's showing that, so for example, on the blue one in the bottom right, how the violin is shaped, it's a little larger on the top. Because all the individual data points are not there, that's just indicating that there's a lot of dots up there and less as we get closer down. So moving into scatter plots, these display individual data points on an X and Y axis. So on the top, you can see the X axis is JAK2 VAF, or the allele burden of that point mutation, the V617F point mutation in the JAK2 gene. And the Y axis is their normalized read counts for IL-1 beta. In this case, that's just a gene that they did sequencing on. And what they're trying to show in that top plot is that as the allele burden increases, so does the IL-1 beta in the bone marrow. And to show that, you have that black kind of diagonal line, and that's an R or an R squared value. The closer to one that gets, the higher likelihood that we have a direct correlation. It's just a mathematical tool to try to show if something is directly correlated or indirectly correlated. Another type of scatter plot, just a line plot, again, X and Y values, but you can connect the dots. Nothing really different there other than dots are connected. And usually when you are showing a line plot like that with connected dots, the x-axis is time. And then the last one that I think is probably the most familiar is a survival curve where the y-axis is survival and the x-axis is almost always time. The y-axis in this case is a percent, but times 100. So 1 is 100%, and then that zero point 0.8 is 80%. And you all know and are familiar with how to read these, whether it be a survival curve from just a disease like this is, or if you start including different drugs, you have a control group or a group that received the drug, and you are looking to see if there's any kind of survival curve. This is, of course, very, very common in clinical reporting of outcomes for clinical trials. Okay, so now getting into a little more of the mystifiable data. This is a volcano plot on the top, and obviously called a volcano from the shape. No one expects anyone to be able to look at this and know exactly which of those words up there, in this case, these are genes, are what the author or presenter is talking about until they directly call it out. But the things that I do want to point out, because in every volcano plot, there are certain things that are going to be consistent. In each volcano plot, there's always two comparisons. So in the top left, you have reticulin grade 0, so no reticulin, fibrosis in the bone marrow, compared to reticulin grade 2, so someone who has a grade 2 of reticulin fibrosis in their marrow. And all they were trying to do, I'm not sure what cells were used here, but this is just a comparison and sequencing of genes, and you want to know what's up and what's down. So the lines that are going up and down are fold change. So everything to the right or in the red is statistically elevated, and everything that's down and on the left or in the blue is statistically decreased. The line that's going this way is that p-value that we were talking about. So not only is it elevated and it's 2, 3, 4, 5 times higher, it's also statistically significantly higher. Everything that's higher is going to be in the top right. Everything that's lower is going to be in the top left. And all that black that's down at the bottom is either not a p-value that is not significant, or it didn't have a significant fold change. It's a common visualization of sequencing data, as I said, to show dysregulated genes. This can be RNA-seq, this can be DNA-seq, this can be different times of sequencing for protein. All of that, of course, the presenter should be able to walk you through. And then heat maps, often, again, this would be taking every single dot in one of those volcano plots and trying to put it onto a heat map. And they can be simple, like the one on the bottom left, or they can be really complex, like the one on the right. And the one on the right is thousands and thousands of genes. But instead of labeling the genes, they've labeled pathways. And that's a very common way, especially in MPN research, putting these heat maps up to walk through certain pathways to show what's up and what's down. Again, red being up and blue being down. And the last one, again, pretty complex, is a UMAP, and this is a uniform manifold approximation and projection. So I think the first piece of advice is if anyone ever calls it that, it's going to be a very complicated presentation. Everyone just says UMAP. And what this does, this is millions and millions and millions of cells from bone marrow that you pass through a machine, and it counts them, and it's counting them based on what's happening on the outside of the cell. It labels B cells, it labels dendritic cells, it labels mast cells, and it tries to put them in a shape. The shape is totally abstract. The shape does not mean anything. But what it does is it provides a map. So the top one is a shape that, again, doesn't really mean anything. It's just clustering things based on how similar they are. To then in the bottom, when I'm looking at, in this case, it's TET2 wild type or TET2 mutant cells, the progeny from those. You can see that in the TET2 mutant progeny, there's way more of that bottom left kind of red color. So there's a lot more of those cells being made in the TET2 mutant than the TET2 wild type. It's a very simple way to think of a UMAP. That's really all that they're trying to show is how things are similar, cluster them all. And then when I look at my different genotypes or my different mutations, we're able to see what types of cells do certain things based on whatever question you're asking as a researcher. Okay. So moving into a example of a good and a bad slide. So this next one is a really, really bad slide. And it's something that honestly, when presenters get up and show something that looks like this, it's not on you at all as a person in the audience to look at this and have no idea what's going on. Uh, Obviously, Dr. Moreno made this. But unfortunately, this is not just make this as bad as possible. Nothing could ever look this bad at a presentation. And unfortunately, they do. And so I guess what we're trying to, and what I'm like begging you to understand is this, looking at this and saying, I don't get this. This goes way over my head. I don't understand what's going on. Again, not on you at all. This is 100% on the presenter if they show something like this. So a couple quick things I want to point out. This title makes absolutely no sense. It does not follow the rules of a title that I was laid out for you of what a title should be. You have different fonts, you have different types of graphs. Nothing is labeled appropriately. And something that I want to point out from a patient perspective is Dr. Moreno put stable disease and certain death. And obviously, that's a little bit of an exaggeration. But so often, there's such sensationalist and overdone words that scientists will put on presentations that really shouldn't be said, especially knowing that there are people in the audience listening to this that either say, "Oh, that's going to cure what I have," or they're going to say, "Oh, I have that." Like, and you're saying that 100% certainty that it means that I'm going to have a shortened life and I have 6 months to live. I think both sides of that equation are wrong, and a presentation should never be that. This is a good slide. So here it's very clear. The title is very clear what the takeaway is. Every acronym is defined, and there's not 50 different panels on this slide. As a presenter, you can press forward as many times as you want to. So there should be less information on each slide that's digestible and easy to understand. Okay, so this section is a lot shorter, and this section is trying to convey how scientists evaluate findings across MPN research types, meaning preclinical, basic science, clinical, what translational science is. You hear that all the time. Just trying to demystify those terms for you. So going from left to right, this is time, if I was to put an x-axis here. So you start with basic science, which is understanding fundamentals of a biological concept. Mitochondria is the powerhouse of the cell. That is a basic science. We know this. We're taught this in high school. But without knowing what mitochondrial function is, we would not be able to study what happens with mitochondrial dysfunction in cancer. So moving to preclinical, this follows clinical observation or reverse translation. I'll define that in a second. And it defines mechanisms and discovers disease-modifying targets. It tests in various models and systems, and you move from preclinical often to clinical. And that's the bench to bedside or the translational approach. And I said reverse translation because a lot of times you'll find an issue in the clinic and you will try to reverse translate that and try to cause that in a lab setting to be able to understand what's triggering it. And if you're able to make a model of something that happens in the clinic in real people, you're a much higher likelihood of finding some kind of target, whether that's a drug target or just a biochemical target to try and undo what's being dysregulated in people. But moving from preclinical to clinical, so now clinical research is more about evaluating safety and efficacy. These are your clinical trials that occur in 4 different phases. It's very outcomes based and it's often prospective. We're tracking things as they happen in people. And then, of course, after a successful clinical trial, it gets put into practice where it's adopted and adapted into clinical care. Think for MPNs, ruxolitinib. So this would have went through this entire process, and now it's kind of a very, very common, almost the most common drug for MPN patients. Preclinical work takes a very, very long time. We have decades of work that goes into trying to find something that can move into clinical trials. And I know all of you are aware clinical trials also take a very long time. But something from, again, a patient perspective is if it's unclear which of these boxes we're talking about while we're giving a presentation, it's totally reasonable for you to ask, where are we at? And what are the next steps? One of the last things that I want to discuss before I give it back to Dr. Moreno is talking about the models and limitations of preclinical research. So, of course, I, as a scientist, I occupy this preclinical research phase, and where all of my work is done is, or 90% of my work is done in a preclinical setting. I just want to kind of talk about what that means. Most of the presentations that you see are going to occupy this space unless it's a clinical trial. So translating biology into clinical trials and treatments requires incremental work. Like I said, this can take over decades of time, and it's meaningful even if it's not definitive. And I think that's really important. A strong signal in a small study a biomarker change that supports a mechanism or safety data that enables a larger trial, they're all meaningful. And limitations are not weaknesses. I think it's very important for a presenter to list the limitations of their study. And as an audience member, that does not mean dismiss what they say. If anything, that means they've put a lot of thought and consideration into the limitations. That makes it really important. So there's three main types of preclinical models that I want to quickly go through. The first is in vitro or ex vivo systems. You'll hear done in a dish. That's what this is talking about. Highly reproducible. You're using cell lines. It's often an important first step because if it doesn't work here, it's not going to work in animals and it's not going to work in people. But it's also the least time consuming and the least cost restraint. And then you have in vivo systems, and a lot of time this is where those decades of work happens. And this is, I have the word murine there. If you're not familiar with that term, a lot of times we'll use that term to talk about mouse because PDXs, which I'll talk about next, are also done in mice very often. So we'll say a murine system or a PDX system, and I'll talk about that, why that's distinguished in a second. But now we're getting into research, a research system that's maybe a lot more relevant for MPN. You can study niches, you can study fibrosis, you can study variant allele fraction as you have hematopoietic or blood stem cells that are recapitulating and repopulating a living environment. You can cross mice to induce specific mutations, like the point mutation in the JAK2. You can do that with high precision. If you want to test a low VAF, a lot of patients listening probably have a VAF of 10% or lower. A lot of patients listening probably have a VAF of 60% or higher. And it's really important to not treat those the same way in our experimental systems. And by using a murine system, we're able to really calculate how much of that is going into each of our systems to ask our very pointed questions. And then the last section are PDXs, and this is patient-derived xenograft. So instead of crossing mice and breeding mice to get mice that have a certain genetic mutation, we're able to transplant stem cells from a patient that donated to a research study into mice that don't have an immune system, so they don't automatically reject the cells. And you're able to create an MPN environment in a system that has a humanized blood system. So then you can use human drugs to treat the disease. And it's kind of a step closer, a half a step closer to a clinical trial. 99% of clinical trials in 2026 are not going to launch unless what was done in a dish and in your murine models have also been done in a PDX. And this is my last slide. So this was just a very quick example that I found walking through this entire process. So the basic science of this Nature paper was that there was a new strain of cave bacteria that was found. And at first glance, you see that, and there's no way this has anything to do with cancer. But then, well, the machinery differs a little bit than how we typically think of CRISPR. And in a dish, it showed selective killing of the cancer cells. So the cells that had a specific mutation, we were able to selectively kill them. Okay, so let's move it in vivo. And in vivo, we actually shrunk the tumors significantly. This is a huge, huge step, but we're still so far away in this of trying to figure out if we can make this a clinical trial. And that step right there is that decades of research that still needs to happen. This is extremely powerful and this is extremely helpful because all these preliminary findings, whether that preliminary finding was finding a cave bacteria that many people would look at and say this has nothing to do with health, to finding that it differs in its machinery and the way that it works, to seeing that, well, actually, we might be able to use this for cancer research. And each of these iterative steps takes us closer and closer to a bench to bedside. And the last thing that I have on my slide, and I've been saying it the whole time, do not overstate things. And when they do, maybe as an audience member, look at that and say, maybe I believe a little less of this now. If someone says we have found a cure, they probably didn't. And I think both in the preclinical research and the clinical research, this happens a lot. But I really don't want to downplay the importance of each of these steps and how each of these steps is crucial to this bench to bedside research thing that we've been talking about. All right. So now that I have given you a ton of new information, it is now my co-host's job to give us a couple things that we can take away immediately and try to apply this as MPN researchers as we and MPN patients as we try to understand better what scientists are saying when they present.
Thank you, Dr. Parsons. This is our final section. I'm going to offer some practical tips for immediate use as you explore the world of science and medical information. During presentations, it's helpful to not dwell on the words or concepts that you don't understand. Doing so might prevent you from hearing other information, making you fall further behind. So here's a trick I learned in grad school that I call holding mental space. I hope I can do a good enough job explaining it. As you're taking in new information, building your knowledge, sometimes you won't have all of the background you need. So let's say I'm reading this abstract and there are unfamiliar terms or concepts represented by the black spaces. If I'm just a little mentally flexible, I can read past the black spaces and try to understand the sentence or the abstract in general. It's like learning a new language. You might miss some words, but if you could be comfortable holding that space empty, it allows you to move forward and understand what you can. Once you have the overall context, you might be able to infer what those black spaces meant. So it's important to not get flustered, to stay present, move forward with what you understand. And remember, it's not a test, so you can look things up later. But if the amount of mental space you're holding during a presentation starts to look more like this, there's a problem. This is dense, expert level content. Returning to the language analogy, I wouldn't take an English 101 class to see Macbeth, just like I wouldn't give this abstract to a newly diagnosed patient. When stretching your mind becomes stretching your sanity, remember, in science, confusion is normal. Let go and find some more approachable content. You might wonder after a presentation what that information means for you or the field as a whole. Here's how to find those takeaways. Your anchor should be the key points or conclusion in the presentation. And thank goodness a lot of presentations now include an explicit slide titled Key Conclusions or Takeaways. Still, they can be pretty dense, like this example here. So here's a tip. Have your phone ready to take a picture of these slides. Or if you're watching on your computer, memorize the screenshot keystroke so you can capture these takeaways instead of attempting to furiously type them and not be able to hear anything that's being said. Another tip is please switch to digital notes. So much of how science is presented now is digital. It's easier to copy a slide picture into a digital document than it is to do that into a physical notebook. Another tip is to keep your notes in the same format, whether that's Word documents or Google Docs. There are also some programs that you can use like Evernote or OneNote. This will allow you to search for your key terms across all of your notes. Good science communication tries to make it clear why what is being shared is important, but sometimes that meaning still isn't clear. Maybe you sat through a presentation all about mice and you thought, so what? Well, remember what Dr. Parsons said, science builds on itself and basic science matters. Today's preliminary data might be the basis of clinical practice tomorrow. And remember, learning is tough. Maybe the talk isn't at your level or it's on a different kind of cancer. During your next challenging presentation, see how long you can follow it. Maybe the takeaway is the experience of stretching your brain. Building your knowledge is never a bad idea, and it can help you understand more in the future. But remember, you have my permission to walk away from any presentation, except this one. So the presentation is over. You didn't walk out, and you'd like to ask the presenter a question. First is to ask yourself, is this the best person to ask? Then follow these 4 rules for how to ask clear, clarifying questions. First, thank the speaker for their time because manners. Second, refer to a relevant part of that presentation. Third, ask your question in 2 sentences max. If you can't ask your question in that amount of sentences, maybe you need more time to think it over. And finally, if something wasn't clear in their answer, it's a good idea to just follow up later via email. All presenters include their email or how to reach them or somebody else that can get answers to you on their slides. And you want to make room so that other people have an opportunity to ask their questions. So here is a act out to describe all of these 4 things being done in one question. So I'm at a presentation, there's a doctor in front of me, and so I say, "Thank you, Dr. Seuss, for coming to speak with us all today. I was curious about what you said regarding the 1 fish and subsequent 2 fish. Are we to understand from the illustration that it is in fact an additional 3rd and 4th fish that are the aforementioned red fish and blue fish?" Every conference audience knows what bad questions feel like. Here is what will make a presenter and an audience cringe. If the question is off topic, if the person cites their personal lab results or medical information and asks for medical advice, if the question goes on and on past the 2-minute mark, 2 minutes is what the presenter probably had to defend their hypothesis. And now your question is going longer than that? Bad form. And finally, the worst mistake made by beginner and experts alike, the question that's less of a question and more of a performative, "look what I know" comment. So to illustrate this type of bad question, here is another act out. "I have a medical question for you, Dr. Seuss. But first, I've eaten green eggs and ham and would like to tell you my harrowing tale." These are important lessons that you can take with you to make sure that you're the best audience member you can be. So, what about our takeaways? The first thing you need to remember is science is hard. Even the most brilliant scientists or researchers don't fully understand everything. We're all struggling together. So it's okay not to understand complex science. It's a puzzle and it's not even done yet. And nobody, let me reassure you, nobody is more understanding about not understanding than a scientist. It's what we're used to. We want to answer your questions because sometimes they're our questions too. The second thing that I hope you took away and understand is what to expect. We covered a bunch of different basic structures of science presentations and their components. We talked about different science questions and how data can be presented. And third, I hope you see that that pattern that we taught you can prepare you for how science is going to be presented to you in the future. Knowing the pattern or landmarks, as well as having a willingness to learn, will help you in any science presentation. And with that, thank you so much for joining us today as we did our presentation on presentations. Dr. Parsons and I hope you're better equipped for the scientific content in your future, and we're happy to answer any of your questions now.
Wonderful. Well, thank you so much, Amiel, Dr. Moreno, and Tyler, Dr. Parsons. It was such a great presentation and super helpful as, patients and even myself go into these scientific presentations. They are very complicated. I know we're at time, but while I have both of you here, I am going to ask you a couple questions because I would hate to miss the opportunity. The first one is a great question is, what is the best way to explain something I read or I saw in a presentation to my care team? I want to bring that information to my care team as this great new research I saw. How do I translate that to my care team to better understand that together?
Tyler, would you like to take this one?
Sure. I think we know a lot of stories of patients bringing in a booklet of 10,000 printed pages for a doctor's appointment, that there is 10 minutes that they might have. And then I think we also have a lot of examples of patients who might bring, as Dr. Moreno pointed out, the abstract for a certain study that was going on. Or I think what we're seeing a lot in the last couple of weeks is, hey, I saw this thing at ASCO and it looked very promising. There were a lot of MPN clinical trials presented at ASCO. And I think very, very common. Your doctor probably has heard of it, or if they haven't, they know exactly where to find the information that you are bringing up to them. I think that I have never spoken to a doctor who dislikes this type of conversation with the patient. And I think it's really important. I think that bringing in 10 or 15 studies is probably not the right move. But I think bringing in, whether it's a printed abstract or messaging them in MyChart, I know a lot of patients have had their ability to kind of reach out and contact their care team through MyChart, and they have sent studies that way as well, which I know has worked, and I think might give the physician and the care team a little more time to digest what's going on when it's not immediately during a patient care window.
Amazing. Thank you. And another question, if I wanted to just start following research, where would you recommend I begin? Amiel, that seems like one for you.
Well, sure. Well, how I follow research is by having a Google alert on my on set for keywords that I'm interested in. So I might put in polycythemia vera, and Google will search on daily to see if there's any new content put online about that. It's a large net, so you can get a lot of different content, both beginner and expert level. But through there, you'll be given a bunch of material. Then you get to decide, once you go and visit those individual sources, which ones are writing for you, which ones are things that are matching your level of understanding, and you think that you can gain the most insight from. So that's a first step in order to find a lot of content that then you get to decide what you want. Does that answer the question?
It does. Thank you.
Great.
Yes. And, Amiel puts out great work at the MPN Research Foundation. So
Oh,
you're already following and getting some of that research through your communications. So that's great as well. There was another question on what are your thoughts on using AI to parse complex papers to gain a more simplistic or digestible overview?
I think AI right now is limited in its ability to accurately deduce a correct summary from some of those more complex papers. Sometimes if you have the ability to build out your AI agent, you can give them more than one paper. Dr. Parsons mentioned review papers. So the AI agent is a naive thing that's going to attempt to learn. Give it a bunch of information to learn from before you expect it to be able to accurately provide you accurate answers. So what I would do is I'd find several review papers that were cited in one review paper that I'm curious about and load them all in at the same time, and then have it try to provide some context for me. Even so, it's very dangerous. I often find that AI is incapable of correctly discerning things correctly when they're going from very complex to simplified. That direction can be a struggle still.
Okay, thank you. And then, I think we've answered this, but is it ever even appropriate for a patient to ask a question at a scientific conference? Is that encouraged, or what are your thoughts around that?
I
For
think it
sure.
I think it highly depends on what the question is. I think if it's about the presentation, extremely welcome. I love that because I'm being asked a question that I likely, if it's about my presentation, have an answer for, and that always feels good to be able to answer a question. And to know, I think any scientist who is not appreciative of the anyone in the world who can benefit, who can stand to benefit from the research that they're doing, asking questions of the research, I think is in the wrong profession. And I think having a question from the audience member who is a patient ask again, if it's about the presentation, it's probably not appropriate, as Dr. Moreno said, for a patient to go up and ask, is this applicable to me? Because here are my numbers. That's not probably the right forum to do that. But I think if it's a question that is at at a forum where new work was presented and it's about what was presented, extremely welcomed.
Great. Well, thank you both so much for this incredible presentation and for spending some time answering these questions. I know if anyone else has questions on, and you're still here, please email us. I work with Amiel every day, so we'll make sure those answered. And we've got Tyler on speed dial, so we'll always know how to reach them. But thank you both so much. We're going to finish with just a few housekeeping items. Um, Again, we want to share that the MPN Progression Registry is open for enrollment. So if you are an adult in the U.S. living with an MPN, you can play a direct role in advancing MPN research. Join today by visiting mpnresearchfoundation.org/MPN Progression Registry. Or I think Emily is putting the link in the chat, and then also there's a QR code if you are able to scan that. The slides will also have the link if you can just open those, the PDF that we provided. And then this slide introduces you and MPN. So this is a patient and caregiver education resource developed by MPN Research Foundation. We've designed this program to empower patients and caregivers with knowledge. On the website, patient and caregivers will find animations, slideshows that really explain complex concepts in simple terms, expert physician videos that answer these common questions, patient stories that shared lived experiences, and infographics, quizzes, and downloads that make learning more interactive. And you can just visit that by visiting unmpn.com. And then we're very excited to announce the next session. So join us for the final MPN pathway session featuring Dr. Ruben Mesa. What a way to end this series. He will explore the lived experiences of individuals with MPNs, highlighting the real-world symptom burden and quality of life challenges faced by patients and caregivers. This session will also emphasize the importance of patient-reported outcomes, or PROs, in improving communication, personalizing care, and advancing research, including their use in clinical practice and the MPN Progression Registry. Dr. Mesa is a leading MPN expert, I'm sure most of you know, and a contributor to multiple FDA-approved therapies. He's going to share these insights into symptom management, key PRO tools, and strategies to better address patient needs. So join us Thursday, July 27th from 4 to 5 p.m. Central to learn more about this. And then we also have an exciting MPN United webinar series. So this is led by our partner, the MPN Advocates Network. We're excited to share an upcoming session focused on patient advocacy and access. So that title is Advancing Access to Strengthen Patient Advocacy and Access Initiatives. This will take place on August 25th. From 7 a.m. to 8:30 a.m. Central Time. But we are really trying to reflect our global audience, and so it's designed to really accommodate some of our participants in the different regions. And just again, as a reminder, the evaluation of the session will be sent to you in a post-event email or in the chat there. So please be sure to complete the evaluation for this session to earn your credit and just to give us a better idea on ways to improve or what you really enjoyed. And with that, have a wonderful rest of your evening. Thank you for spending some of it with us, and we'll be in touch.